Thank you very much, Jill and Wayne, for inviting me to present in this E3SM webinar. Today, I will be presenting work from that I have been working on towards improving crop representation in ELM. I would first like to thank my various collaborators across the DOE lab, Kate Calvin, Ben Bond Lamberdy, Beth Runiak, Dan Ricuto, Hachik Sargason, and Gautam Bisht. Over the past several decades, human activity has significantly altered the land surface. Large demand for food production has resulted in almost 11% of our global land being used for agricultural production. And agriculture itself has can alter the regional as well as global climate. And the reason it does that is because agriculture has a lot of biogeochemical and biophysical impacts on the climate. For example, when we replace native vegetation with cropland, it results in the emission of large amount of CO2 gas to the atmosphere. Agriculture itself has been shown to be actually a net sink of CO2 during the peak growing season. However, certain agricultural management practices such as tillage and the growth of certain crop types can result in agriculture becoming a net source of CO2 and other greenhouse gases such as methane and nitrous oxide. Because of these biogeochemical impacts, as well as biophysical impacts of modifying the surface energy budget and the water budget, agriculture can alter the regional and the global climate. And therefore, Earth system models that are used for, that are developed for projecting the climate, we need to have adequate representation of crops or agriculture within them. However, most Earth system models still have very simplistic representation of crops in them. For example, they will represent crops as grassy crops rather than identifying various types of crops. And this ignores the various, this misrepresents or doesn't really capture the various phenological stages of the crops and the differences across the crop species. And it has been shown that adequate representation of crops actually has a large impact on carbon fluxes. For example, the figure here shows the difference in the maximum monthly average GPP when crops were adequately represented, various crop species were represented, and agricultural management was represented, versus crops being represented as grassy crops in the CLM model. And we can see that the GPP is grossly underestimated in various parts of the globe. And this shows that we need to have proper representation of crops in our Earth system model. Apart from representing crops that are currently being grown a lot, like cereal crops across the globe, we also need to think about crops that may actually increase in the future or may be grown a lot in the future. For example, we know that in the future, increased demand for energy and the need for mitigating the impacts of climate change, there will be large increase in bioenergy production. And this figure is showing an example of that. This shows the projected increase in bioenergy cropland area across the 5 different SSPs till the end of the 21st century. So almost for all SSP, we are seeing large increases in bioenergy cropland area. And we need to understand the impact of such large increase in bioenergy cropland area on various energy fluxes, carbon fluxes, as well as the water budget. And in order to do that, we should have adequate representation of bioenergy crops in our Earth system models. So with that in mind, the main objective of my research was expansion of ELM crop model to include perennial bioenergy crops. Now in ELM, after the addition of perennial bioenergy crops, we also need to think about what parameters to use for these bioenergy crops. And the reason is that because currently the crop models in ELM requires more than 100 parameters. And most Earth system models utilize default global parameter values. And this results in underestimation of biases and fluxes that are estimated by using those default parameters as compared to the observed fluxes. So in order to resolve that issue, we try to optimize the parameter to find the optimum parameter range for different crop species for different geographic locations. However, optimizing the parameters is computationally prohibitive due to the model's complexity and the large time it takes to reach biogeochemical equilibrium. To resolve these issues, the prior studies have utilized parameter values based on observations, or they have utilized one-at-a-time calibration. However, both these methods fail to account for parameter interactions. So in our study, we address both these challenges. The first challenge is of actually optimizing the crop parameters despite the computational cost, as well as accounting for the parameter interactions by utilizing surrogates and by first developing ELM surrogates and then utilizing the surrogates for sensitivity analysis and for calibration of various parameters. So in ELM, currently we already have the annual crops. And for addition of the perennial crops, we had to add a new module for the phenology because the phenology of perennial crops is quite distinct from annual crops. Perennial crops have three distinct phases in its phenology. First is the planting, when the planting occurs, which is currently determined based on reaching a temperature conditions. Then the leaf onset occurs after a couple of weeks based on when the growing degree days criteria is met. And after the leaf onset, the leaves and the stems keep growing. In perennial crops, there is no grain, so there is no grain stage or grain fill stage. And eventually, the leaf senex occurs later in the fall when temperature falls below certain criteria. So we added a new module for perennial crops phenology, and we also made few edits to how yield is estimated for perennial crops and how carbon nitrogen allocation occurs. After the addition of perennial crops, we then had to optimize the crop parameters for these crop types. So for that, we utilized the uncertainty quantification toolkit developed by the Sanya lab, and we developed surrogate models of ELM runs. To summarize, the steps that we took for this calibration were, first, we identify out of the really large number of more than 100 parameters, we identify the crop parameters that are most important for perennial crops. And we also identified the approximate range for those parameters based on literature review. And then we generated a sample file containing a large sample of randomly distributed parameters within their specified range. And this sample file was then used for making ELM runs. And an OLMT, or the offline land model test bed developed by Oak Ridge National Lab, was utilized for submitting, managing, as post-processing a large ensemble of runs. In our case we made almost 2,000 ELM runs. We then developed surrogate models for these ELM simulations, and those surrogate models were used for sensitivity analysis by estimating the Sobol indices and identifying the most influential parameters. And finally, we utilized the surrogate model in conjunction with observational data for optimizing the various parameters. So now I will show you some results of our calibration process. So first, this figure shows the GPP values for miscanthus crop. The observations are shown here in light gray and black color. The observations were collected at the University of Illinois Energy Farm. And the light gray shading is daily GPP values over the course of 8 years, or the calibration period. And the black line is the mean across those calibration years. The light orange shading in the background, or the yellow shading in the background, is representing the output of the ELM ensemble. So we ran 2,000 ELM runs, with those parameters randomly varying. And each ELM output, for each of the output, we took the average across the last 10 years. And those values are being shown here in this orange cloud. And the important thing to observe here is that the ELM ensemble captures the seasonality, the leaf onset, the fast increase in the leaf in the GPP values during the early summer months, the peak values, as well as the leaf senex, when the leaf senex occurs and how the leaf GPP falls down. So it captures the seasonality and the peak magnitude very well. And now we have to identify the subset of parameters, or the parameter ranges, that will actually bring us very close to the observed GPP values. So for that, we developed surrogates of ELM, and those surrogates were utilized for sensitivity analysis. So sensitivity analysis helps us identify which are the most influential parameters out of a given large number of parameters. So we started with 20 parameters, but the sensitivity analysis helped us identify the most influential parameters. So in this plot, the x-axis is showing the day of the year, and the y-axis is showing the sensitivity of for GPP, the sensitivity of various parameters to GPP. And we see that out of the 20, only 5 or 6 parameters are actually most influential for the GPP across the entire season. And those parameters were associated with stomatal conductance, specific leaf area, leaf carbon nitrogen allocation, and parameters associated with phenology, such as when the leaf onset can occur and when the leaf senex occurs. So we then used only these 5 parameters for our calibration. And here are the results of calibration. So again, I'm showing the GPP values for miscanthus and the daily GPP values. The black line is the same as shown in the past, which is the mean observation, mean across the calibration years. The red shading is the prior that was used for the calibration, and the prior is basically coming out of the ELM surrogates. And the posterior is after calibration. What is the calibrated value? Basically, based on the optimized parameter ranges, what is the calibrated value? And as you can see, the calibrated value, the green shading, is very closely captures the leaf onset as well as the leaf senex. It also captures the timing of the peak GPP. However, it does underestimate the peak GPP magnitude itself. So we are achieving a lot of capturing a lot of observations like the seasonality, but maybe slightly off in the magnitude. Apart from GPP, we also calibrated ecosystem respiration for miscanthus, which is shown in the bottom plot here. The top plot is the same as in the previous slide. The bottom is the new plot. And the calibration results in really very good fit for capturing the observed ecosystem respiration. And we also did this calibration for another perennial bioenergy crop, which is switchgrass, which is also, data was also collected at the same University of Illinois Energy Farm. And again, overall, we are able to capture the seasonality and the magnitudes. At least we are quite close in the observed peak values for both the crops and both the fluxes. Apart from carbon fluxes, we also calibrated the energy fluxes, but I'm not showing the results here just to save some time. The calibration resulted in, calibration basically identified the optimum parameter ranges for the most influential parameters, which is listed here. So I just wanted to give a glimpse of that. And these parameter values were then used for validating the model. So we performed model validation by utilizing those optimal parameter ranges and running ELM, just one more ELM run based on those optimum parameter ranges. And in these figures, the black lines are again the observation, but now only for the validation years. So we saved 2 years of data for the validation. And the red is the mean across the last 10 years of the ELM run. And I'm also listing, for both GPP and ER, I'm also listing the relative root mean square error between the simulated and the observed values. And overall, we are doing a decent job of capturing the seasonality and the peak magnitude of GPP. For ecosystem respiration, the model is predicting a little higher values. When we look at the switchgrass, the results again are quite reasonable and much better for ecosystem respiration. And one thing to highlight here is that there are, of course, annual variation as well, and it's highly likely that the ER values during the validation year for miscanthus were slightly different than what was used for calibration, which could have resulted in lower validation for this flux. And I forgot to mention one thing. We calibrated 4 fluxes for each of the crops, and we did that in a combined manner, which means all 4 fluxes, GPP, ER, sensible heat flux, and latent heat flux, were calibrated together so that we end up with one set of parameter for each crop type. Apart from validating the fluxes, carbon and energy fluxes, we also validated LAI values. So now the plots, the left-hand plot is showing the validation results for miscanthus, and the right-hand is showing for switchgrass. The dots are showing the observed LAI over the validation years, and the black line is showing the average LAI over the last 10 years. The model captures the difference across the LAI between switchgrass and miscanthus. Switchgrass has higher observed LAI, and model does capture that correctly. However, it underestimates the peak LAI values for miscanthus to some extent. But we are not completely off. We are still in the same magnitude. We are quite close And overall, switchgrass is better. I would say better validation results are better for switchgrass than for miscanthus. We also validated the harvest or the yield magnitudes. So now this figure is showing the harvest on the y-axis. The red dots are the observed yields for the validation years. So just two values for each year for each of the crops. In the box plots are showing the distribution of annual yield over the last 10 years of the ELM run. So for switchgrass, the observed yield is very comparable to the simulated yields. However, for miscanthus, the simulated harvest is much lower than the observed values. And we did explore why was this happening, because we are doing very good job of calibrating the fluxes. And what we found was that in the field that we are have collected the data from, miscanthus is harvested very late in the season. It is almost harvested in February and March of next year, whereas in the model, the way it has been set up right now, it actually is harvested in November sometimes. So almost 2 to 3 months earlier than the observed. And during this, and most likely this difference in harvest date is resulting in much lower yield, because in case of perennial crops, it's the entire stem and the leaves that are harvested. So most likely that is contributing to the lower observed yield values. So just to sum up the main takeaways, we implemented perennial crop modeling in ELM, and carbon and energy fluxes were most sensitive to parameters associated with stomatal conductance, specific leaf area, leaf carbon nitrogen allocation, and phenological parameters. The simulated carbon fluxes captured the observed seasonality and matched the observed magnitudes closely. However, for miscanthus, the simulated harvest occurs few months before the observations, which results in a reduced or biased yield values. And this work lays the foundation for future work that can examine the impact of bioenergy crops on climate itself, and especially utilize those SSP scenarios with large increases in bioenergy cropland area and see how that will impact climate. So the work that I've just shown was focused on improving a representation of specific crop types in ELM. Apart from various differences across crop types, in agriculture, various management practices are also used or practiced across the globe. For example, crop rotation is practiced globally, and this is done for increasing the yield and for reducing the need for fertilizers and pesticides. The figure here shows locations globally where transition from C4 annual to C3 nitrogen fixing crops occurs. And just to zoom in for the US, most portion of the US Midwest actually have rotation, and most of the rotation actually happens between corn and soybean. And because this is practiced globally and literally occurs every year in most places, and it impacts the carbon and the energy fluxes, because corn and soybean have very different phenology. We need to capture these type of agricultural management practices in our in ELM. So my current work actually focuses on implementing corn-soybean rotation and calibrating and validating the model using spatially varying observations. And this is because corn and soybean, there have been several study sites across the US where they have been collecting data for corn-soybean rotation across the Midwest. So we wanted to utilize data from more than one locations for validating and calibrating the model. So crop rotation was implemented by utilizing dynamic land unit to implement crop rotation in ELM. And for calibration and validation, we utilized data from 3 sites located in Nebraska, Minnesota, and Illinois. And I will show you the results of the calibration itself. So we again followed the very similar methodology as for the perennial crops, of first identifying few crop parameters which are most important, then making ELM simulations, and finally performing sensitivity analysis to find the most influential parameters, and lastly calibrating the model. And the results here are showing the final step, the calibration results. I'm showing the results for corn growth in the Nebraska site, and the results are again for GPP. The black is the observation, and the green line is a posterior. So the model is doing a very good job of actually capturing the seasonality and the peak magnitude of the observations at this site. Apart from corn, we also calibrated soybean for the same site. And you can see just by comparing the 2 plots, just looking at the black flies observations, the soybean has a very distinct, much lower GPP values and sort of seasonal shift. The leaf increase in the GPP, the way it occurs is very different than that for corn. However, the model does capture the differences along the two crop types quite well, and because both the crops are quite well calibrated in the model. We repeated the calibration for the other two sites as well, and across all 3 sites and both crop types, overall we are doing a reasonably good job of capturing the seasonality, the peak values, as well as the differences across the crop types and across the different observational sites. After calibration, we also performed the validation, and calibration was performed for both carbon GPP and ecosystem respiration, as well as for surface latent heat flux and sensible heat flux. And validation was performed for all 4 fluxes as well. And validation resulted in reasonable, relatively low root mean square errors. However, when we validated the LAI, we were experiencing some problems. So in this figure, now the top row is showing the values for corn, LAI, and the bottom is for soybeans, and the columns are for different observational sites. The dots are the observed LAI values for the validation years. And in some places, there were very little observations, so it is missing. And the black line is the modeled LAI values. So across all sites and both the crop types, the simulated LAI is much lower than observed. And this hints at the fact that most likely when we calibrated the model, we are missing some crucial parameters, because of which we are over-calibrating or overcompensating by using some other parameters. So the final conclusion is that I need to go back and revisit the calibration and try to improve the calibration. So my next steps for this analysis will be improving corn soybean calibration to reduce the bias between observed and simulated LAI and yield. I did not show the results for the yield, but we were seeing similar bias, much lower simulated yield as compared to the observed values. Another thing we found was, and which is sort of expected, is that the various crop parameters that we obtained after optimization, they differed a lot across the sites. And this is to be expected because we were seeing the differences in the observation itself, the GPP values, there were differences in seasonality, the magnitudes, and so on. So the calibration is resulting in different parameter values. So the question is, what will be the impact if we were to make regional or global runs? What will be the impact of using constant parameters versus spatially varying parameters? So my future work will explore the impact of constant versus varying crop parameters. And for this, I'm going to be making regional corn soybean rotation simulations for the US Midwest. And I've already generated a land use time series with increased CFTs that include perennial crops and various other crops that have been recently added. And the land use time series also specifies the grid cells where corn soybean rotation occurs in the US Midwest. So now in the last minute, I will just show a small animation of this modified ELM land use time series in which you can see the corn soybean rotation occurring. So on the left-hand side, I have the corn, and on the right-hand side, soybean, a fraction of the grid cell that is corn and fraction of the grid cell that is soybean. And you can see that from the year 2000, there's slight shifting in the fractions. Let me play this animation again. From 2000, there will be slight shifting in the fraction, and that is because some fraction of corn has shifted to soybean in those years, and in the following year, the reverse happens. So this modified land use time series will then be utilized for making regional runs utilizing constant parameters versus spatially varying parameters, and then we will try to see how does it impact the various fluxes, how much impact does it have on various fluxes. So with that, thank you very much, and I'm happy to take any questions.